in 1983, Ashley came to the pediatric neurology clinic. Ashley was the very first child with Rett syndrome that I have seen. But it turns out she's one of many that I would meet throughout the years.
I first heard about Huda Zoghbi's discovery that MECP2 was the gene mutated in Rett syndrome when I was sent a manuscript to review, and it changed our priorities almost immediately. Rett syndrome is a profound delayed onset condition that predominantly, overwhelmingly affects girls.
They will definitely do many things a typical infant will do, as in they will look the parent in the eye, they will babble, they will smile, they will even use their hands in their infancy.
It begins between 6 and 12 months when they fail to meet their normal developmental milestones.
We start seeing regression. Their social interaction will decrease. If they had language, they will lose the words they learned how to say. And instead of using their hands, they will start wringing their hands. They will have difficulty, of course, communicating, learning new things, and they will, with time, develop seizures. They will get spasticity, they will get scoliosis, especially if they are in a wheelchair. And as they get much older, they will get symptoms of Parkinsonism.
It's a sort of constellation of serious symptoms that require almost 24-hour nursing for the rest of their lives. And we don't fully understand why there is that sudden regression.
I was fortunate to meet Ashley, who in 1983 came to the pediatric neurology clinic at Baylor College of Medicine and Texas Children's Hospital. I was really intrigued. This was the first time I saw a child who was born healthy, who did very well, and then lost her skills and deteriorated, but it was not degenerative. I kept thinking about Ashley, and a week after I saw her, it was pure serendipity. A girl who supposedly had cerebral palsy walked into my office wringing her hands. And I immediately recognized all the features of Rett syndrome that I saw in Ashley. And to see two girls with Rett syndrome in 1 week, when no one has reported on this syndrome in the country, it must not be that rare. It must be more common than we think, and there must be other children in our clinic. The second child I saw her in the Bluebird Circle clinic. And I asked the volunteers if they would kindly pull for me all the medical records of girls that had seizures, balance problems, intellectual disabilities. So they came back to me with 35 charts. And as I reviewed these charts, I found additional girls with Rett syndrome. And this is when I realized this is really a real disorder that we have to study and we have to figure out what's going on.
So you've seen all this before, of course. Yeah.
So the question that everybody asked me at the time is, why should it be a gene? This is a disease that's sporadic. Each of these girls were one in a family. And to be honest with you, it was really a strong instinct, gut feeling, backed by one important observation: the clinical phenotype. The clinical phenotype of Rett syndrome is consistent. Every time the girls are born normal, they go through that cascade of neurological symptoms, and always they had the same symptoms. So for me, there's no way an environmental factor will selectively affect girls consistently in the same way. When I moved into my own lab in 1988, I began my work in full force on Rett syndrome and collaborated with Uta Franca and Carolyn Shannon at Stanford. We continued the search and really just systematically brute force going gene by gene, sequencing them. It was 11 years of negative data and very little progress until a person came to my lab. She was a physician trained in medical school, but she has not done her residency. Her name is Ruthie Amir. In August of 1999, the phone was ringing, and it was Ruthie Amir. And she said, I think I found the Rett syndrome gene. I think I found the real mutations. So she came over and we looked at the data, and it was very clear that in all the Rett girls she had sequenced, there are mutations, and that those mutations inactivate the proteins, and these mutations were in none of the parents. Ruthie went back to the lab and she repeated the experiments, and we wrote the paper basically in 2 days.
I was working on DNA methylation. I had found a way of mapping methylated sites in the genome as well as where the genes were. I realized that having this method for mapping methylation meant that I could actually find out whether or not DNA methylation was involved in regulating gene expression or not. At the time I was in Vienna, where Max Bernstiel was starting a new institute, the Institute for Molecular Pathology. What we decided we needed to do was find the proteins that were binding to the methylated DNA. We found one which we called MECP1, but we had trouble purifying it. So we then found another protein using a slightly different assay that we called MECP2, and we managed to purify that. It was pretty clear from the beginning that it's very abundant in brain, and we were able to determine its sequence and show that it bound to methylated DNA, and then began the question of what it did. And our answer to that is that it's involved in optimizing the gene expression program for the entire neuron particularly. And it's that optimization is absolutely essential for the brain to perform all of the functions that it can perform. Huda's discovery that MECP2 was the gene mutated in Rett syndrome opened our eyes to that new way of looking at it. It really led on to our attempts to reverse the phenotype. Having established that the symptoms that you see in humans and the symptoms that you see in mice are really very similar, then you could consider that you had an animal model for Rett syndrome, and therefore you could ask a key question, which was unanswered at the time: is it reversible, is it curable or not? In the case of neurodevelopmental disorders like Rett syndrome, the obvious question is, if I put back MECP2 in an adult, will that correct the problem? That's what led us to do this experiment where we engineered the mice in such a way that they grew up without the MECP2 gene working, because we had put a chunk of DNA into it which messed up its ability to make MECP2. When the animals were sick, we could remove that chunk of DNA and activate the gene. And so now it started to work again. And the question is, did that make things better or not? And the answer was spectacularly that it made things much better. The discovery that Rett syndrome in the mouse model was reversible certainly caused quite a shock that such a severe set of neurological symptoms could be corrected simply by putting back one missing protein.
After the gene discovery, we were really interested in two questions. What are the consequences of removing the gene from brain cells? What are the molecular and biochemical changes? And what are the neurobiological changes? And we began with the simple approach of asking, does this gene affect all brain cells? What are the consequences of taking it from various brain cells? And we were surprised to find that it's actually critical for the function of all brain cells. But because it's important in all brain cells, you couldn't just replace it in one type of cells. We also learned that it is a circuit problem. And it's these findings that have inspired us to think about manipulating the circuit to improve a behavior. In the case of learning and memory, we noticed if we manipulated the hippocampal circuit by stimulating neurons that project to the hippocampus, we can improve the activity of the neurons in that circuit, and we can correct the learning and memory deficit in the Rett mice of that whole circuit and increase neurogenesis, which is normally decreased in Rett syndrome.
We are also interested in the therapeutic side of things. We are looking at using gene editing to correct a specific subset of Rett syndrome patients. The simplest possibility is to put back the gene by gene therapy. So you could put back a good, if you like, MECP2 gene in place of the mutated one. There are complications with this. Too much MECP2 is as bad as too little. And so you need to control quite carefully how much you put back. And with a virus, it's a bit of a blunderbuss approach where you can't control how many viruses go into individual cells. So some may get too much and that may be bad. There is a simpler possibility, which is to edit the genome, change the genome or introduce a new MECP2 gene which replaces the defective one. There are good reasons to believe that that will at least reduce the symptoms and may well cure the disorder to a great extent. The prospects for actually doing gene therapy on humans are quite good. Treatments in humans are planned. I would certainly be very disappointed if it takes more than 5 years for that to be attempted.
Watching girls with Rett syndrome, you feel they're there. They're just waiting for someone to help them get over all these symptoms they're struggling with. I feel there is a great opportunity, and I hope this opportunity will come when one day we can help them.
While I'm still available on Earth, I would love to see someone cured of Rett syndrome, because I'm absolutely confident that there is the potential to reverse many of the symptoms associated with this disease. And I'm waiting with bated breath for that to happen.